What is bioart? How is it defined in contemporary art? Nature has always been a fascination for artists, as we have seen, and as anybody knows. It still is, it always will be, and as technology and science evolves, the palette of the artist extends and expands. With biotechnology, genetics, and technology in general really expanding into our bodies and our environment, the artists have entered into laboratories and work directly with biologists, nanotechnologists, geneticists, neuroscientists, going into the flesh, into the cellular tissue level. And there's some debate how you define bioart. Some artists feel that it's important to be in there working with the tissue directly or with the cellular level directly to be justified to be called bioart. Others think that it's important to have that kind of collaboration with scientists and use scientific imaging, and that would constitute bioart as well. Further, there's more debates going around how animals are used in bioart and what are the ethical implications of artists actually meddling with the genetic structures of natural systems. So let's take a look at some of the artists who have made some perturbations in this field and who raise questions that actually science brings to us. Eduardo Katz may have coined the term bioart, but it is really Joe Davis who was considered and is considered a pioneer. Joe Davis walked into the MIT Center of Advanced Visual Studies uninvited in 1982 and was appointed a research fellow. He already had an idea that genes and genomes can be a new palette for an artist, and started thinking about how to convince molecular biologists at Harvard Medical School and at UC Berkeley to teach him how to synthesize DNA and insert it into genomes of living bacteria. Of course, they were not very comfortable talking to him, and he came up with ideas that scientists would consider dangerous and even crazy. For instance, there was a sequence of amino acids that forms into highly geometric capsules. He had an idea of creating Kepler's nested polyhedra in these viral capsids. The first one is the audio microscope, a microscope that translates light information into sound, allowing you to hear living cells, each with his own acoustic signatures. This is done by quite a few artists since, but he did this very early on. And then he continued with these experiments with sound and cell structures with the next project, in which he looked at how E. coli respond to jazz and other sounds. This he did together with Zaretsky, who is also a bioartist who worked with him in the lab and thought about microbiology and metabolites and antibiotics in relation to sound waves, how to produce sound waves stressful to bacteria. Putting another project is looking at a map of Milky Way in the ear of a transgenic mouse. He took the map of the Milky Way and reduced that information to a sequence of 3,867 DNA base pairs. And then he put it in the transgenic mouse. He thought of primordial clocks, a new way to test the theories of self-assembly. To him, if life could assemble for simple molecules, so could clocks. He looked at the use of high-voltage electricity and space-bound signals and drew up plans for channeling lightning bolts into pulsed laser of unparalleled energy into towering sculptures that would change color and emit tones. So Davis set out about creating an infogene, a gene to be translated by the machinery of human beings into meaning, and not by machinery of cells into protein. His idea was to send a message in a bottle to extraterrestrials, to genetically engineer a sign of human intelligence into the genome of bacteria, grow them up by trillions, and throw them into the heavens, to the land where they may be. For his bottle, Davis chose E. coli, a bacterium on which humans depend for proper digestion, and one that in NASA experiments has survived more than 5 years of exposure to intense cold and radiation of deep space. For his message, he selected microvenus, a simple symbol like a Y and an I superimposed that is both a Germanic rune representing life and an outline of external female genitalia. When I asked him why he chose female genitalia, he said it was his response to all the male phallic symbols being thrown into space, and that he felt that the women are the creators and that he should balance this gender inequality. This is how eccentric and brilliant this artist is. He digitized and translated into a string of 28 DNA nucleotides, microvenus, between the genes of E. coli. The bacteria quickly multiplied into its beakers into billions of cells, each carrying a separate instance of the icon. He claims that he is probably the most successful publisher in history. If you think back into the printing press and then moving into digital publishing, now we're talking about publishing via genetics and genetic engineering. You could see that microvenus is probably the most highly produced graphic at this stage because it is genetically engineered to replicate and go out into space, no less. Microvenus is probably one of the most highly reproduced graphics, but it has never been shown publicly in a gallery because no gallery is willing to risk the public display of genetically engineered bacteria. So the only way we see microvenus is through prints and through the medium that you're looking at now. Recently, a documentary was released on Joe Davis's life, Heaven, Earth, Joe Davis, shows the life of this very eccentric artist who set a precedent for so many bioartists that followed. The question the documentary asks is, why would someone want to encode poetry into the eyes of a fly, fish for a paramecium, or send transgenic organisms into outer space? If we look at some of the artists who followed, like Eduardo Katz, we will see that artists like Joe Davis and Eduardo Katz and many others are really posing important and critical questions about what is life and what does it mean when we manipulate life. Speaking of controversies, we have to mention Eduardo Katz's fluorescent bunny. This is almost an iconic piece for bioart that brings up all the questions that are still relevant, and it is concerning an albino bunny that had genes from a fluorescent jellyfish inserted. So this was created by scientists, and Eduardo used it and claimed it as his art piece. What does this mean? Eduardo Katz named his GFP bunny Alba. And the way it was created by French scientists was by using a process called zygote microinjection. In this process, the scientists plucked a fluorescent protein from a species of fluorescent jellyfish called Aquaria victoria. Then they modified the gene to make its glowing properties twice as powerful. This gene, called EGFG for enhanced green fluorescent gene, was then inserted into a fertilized rabbit egg cell that eventually grew into alba. As the cell divided, the green gene also replicated and made its way into every cell of her body. This isn't the first time a mammal has been designed to glow. In 1997, Tokyo scientists added a glowing jellyfish gene to mice. The mice were created for research purposes to provide animal models for studying biological processes and diseases. The luminescent jellyfish genes can be used to tag certain genes or proteins. When the protein is active, scientists can detect this fluorescence under a black light. When there's no activity, no fluorescence appears. That kind of tracing ability allows scientists to watch the effectiveness of potential drugs as they affect the body without using surgery. For example, anti-cancer genes can be inserted with the glowing genes, so a light source is all that is needed to learn if genetic manipulation is successful. But what happens when an artist decides to use this same exact project and claim it as art? In fact, Eduardo Katz calls this transgenic art. He considered this a new art form based on the use of genetic engineering to transfer natural or synthetic genes to an organism to create unique living beings. But animal rights activists claim that the project is needless and abusive manipulation of an animal, while scientists who work with the fluorescent proteins have pretty much dismissed the project as interesting but silly. Here's what Eduardo Katz says about Alba, the GFB bunny. Alba is undoubtedly a very special animal, but I want to be clear that her formal and genetic uniqueness are but one component of the GFB bunny artwork. The GFB bunny project is a complex social event that starts with the creation of a chimerical animal that does not exist in nature, and that also includes it at its core. Here are the issues that Eduardo Katz lists that are important for us to consider. One, ongoing dialogue between professionals of several disciplines: art, science, philosophy, law, communications, literature, social sciences, and the public, on cultural and ethical implications of genetic engineering. Two, contestation of the alleged supremacy of DNA in life creation in favor of a more complex understanding of the intertwined relationship between genetics, organism, and environment. Three, extension of the concepts of biodiversity and evolution to incorporate precise work at the genomic level. 4. Interspecies communication between humans and transgenic mammal. 5. Integration and presentation of GFB bunny in social and interactive context. 6. Examination of the notions of normalcy, heterogeneity, purity, hybridity, and otherness. 7. Consideration of non-semiotic notion of communication as the sharing of genetic material across traditional species barriers. 8. Public respect and appreciation for the emotional and cognitive life of transgenic animals. 9. Expansion of the present practical and conceptual boundaries of art making to incorporate life invention. So as you can see, this artist may be considered by scientists to have done something frivolous. And he may have created a lot of debates on what is considered life and art and what are the ethical implications of the kind of work that he did. But he also has very thoughtfully laid out issues about his role in a larger society to create this kind of dialogue using his artwork and his conceptual premise about biology, technology, genetics, engineering, and creativity.